We deal mostly with neuroblastoma, that is one of the most frequent solid pediatric cancer, and for which the prognosis is not very good. About 50% of the high-risk patients die, and for the time being, we are all looking for new approaches, including immunotherapy-based approaches, for treating this disease.
Okay. And could you tell us about your research in hypoxia affecting the microenvironment?
Sure. The idea behind it is that if you want to deal with the tumor, you have to deal with the whole tumor mass, which is composed by many cellular types, not just tumor cells, but also leukocytes that infiltrate the tumor, macrophages, dendritic cells, eosinophils, all kinds of leukocytes. And then also you have to take care of the stromal cells, the vessels, so there is a complex mass. Within a tumor mass, you have signals, very important signals. And one of these is hypoxia, namely there is a low oxygen tension, which is very important because every cell in the tumor mass has to feel this low oxygen sensor. There are no receptors, so there are sensors in each cell, and each cell responds to hypoxia. And the response to hypoxia makes these hypoxic patches, areas, and you can find them within the tumor mass, area with highly hypoxic and less hypoxic. Now, hypoxia is associated with the resistance to tumor, to drug treatment, and also improves or increases metastatic spread. So it's a negative factor for tumor. And in fact, we can see in neuroblastoma children, the highest the tumor hypoxia, the worse is the prognosis. So in one way, we have to deal with it, and we have to deal with a situation in which you have non-hypoxic tumor cells and hypoxic tumor cells, non-hypoxic lymphocyte and hypoxic lymphocyte that coexist in different compartments within the tumor mass. And unfortunately, the treatment for those hypoxic tumor cells and non-hypoxic tumor cells has to be different because one cell is sensitive and the other is resistant to treatment. Therefore, it is complex, and there are new promising avenues. I mean, on the one hand, we can take advantage of the fact that the macrophages, dendritic cells, the immune cells travel from hypoxic area to non-hypoxic and vice versa. So hypoxia is not a static situation, it's a dynamic situation. And we can take advantage of that to carry or to deliver immunoreactive cells from one place to the next. Another is to develop drugs. There are now drugs being tested, which are active only under hypoxic condition. So one idea would be to treat sequentially the patients with drugs that are mostly effective under non-hypoxic condition, followed by drugs which would be activated only under hypoxic condition, thereby trying to get rid of the remaining hypoxic niches. So this is basically the leading idea. The problem with that, of course, is that you cannot really biopsy kids sequentially. So you have to find out some way, somehow, to figure out whether we are dealing with hypoxic response, whether the kids respond to therapy. And this is the last approach we've taken, is to monitor exosomes. Exosomes are small vesicles released by the tumors that are floating into the bloodstream. And you can look at this exosome, and the content of the exosome reflects the content of the cells from which they come from. And notably, there are molecules like small RNA, very stable, called microRNA, that can be used as a biomarker of the biology of the tumor. And this is what we've been doing in the neuroblastoma. Other groups are doing similar things in other kind of cancer, big cancers. And there is good indication that, going back to the hypoxia problem, we can derive from these exosomes some signature, some group of microRNA that will indicate whether or not there is an hypoxic area or there is a large hypoxic area in the tumor, and whether or not drugs that should affect the hypoxic area of the tumor are actually working and functioning. I think that the exosome kind of research has the highest applicability because we can do more work on it. I mean, it's a non-invasive way of getting information on a tumor. So even in terms of getting the sample from the patients, all it takes is to obtain 1 or 2 mL of blood, and it's something doable even if we are dealing with small kids. So it's something that can be done ethically and can be done on a sequential basis, so we can follow the response of the patient on time, along the timeline. As you may or may not know, it is difficult to experiment new drugs in kids because fortunately for the kids and unfortunately for research, these tumors are very rare, and companies are not that interested in experimenting drugs on a small population of patients. Therefore, the future of this will depend a lot on what is going on with other kind of tumors. And eventually we can utilize in the pediatric environment tumors that were developed for adult patients or for different. And this is a complication because neuroblastoma is only a pediatric tumor. So we have to rely on drugs that were derived for melanoma, derived for other kind of cancer. But nevertheless, the problem of hypoxia as a risk factor is not unique to neuroblastoma. It's something that occurs in breast cancer, prostate cancer. So it is likely that the research in which you try to deal with the neuroblastoma, with the hypoxic tumor cells, so the hypoxic part of the tumor, may progress. And drugs be developed in other kind of tumors that could be applicable for neuroblastoma.